Hello, everyone. My name is Jen-Hao Yang. I'm assistant professor in Institute of Biomedical Sciences, National Sun Yat-sen University, Kaohsiung, Taiwan. And first of all, I want to thank for EDU Care Taiwan and Lin giving me this great opportunity to share my lab's work and my research direction for my lab. And today, for my research, I was talking about the human skeletal muscle aging, the insight from myogenic RNA binding factors. So basically, we're talking about how we're using RNA and potential RNA drug and to study the muscle aging related function and muscle aging associated process. And in the very beginning, I want to briefly introduce myself. Uh, my name is Jen-Hao Yang again. I received my PhD in National Yang University, Taipei, Taiwan is capital city. And I went to the US for in the NIH, National Institute of Health and Aging Institute for my postdoc research fellow. And I just came back this summer to join the Institute of Biomedical Sciences, starting my lab. I'm very lucky guy. I have a chance to visiting two beautiful harbor city, Baltimore in US, and also Kaohsiung, very beautiful. As you can see here, very beautiful with the harbor and sightseeing. And you must come to see the Kaohsiung city by yourself. Yeah, and it's a very beautiful city. And my research focused on the muscle aging and using RNA as a molecule to study the muscle aging. And talking about the muscle aging, I want to start from this slide. As you can see here, this is our leg section. Like you do this section, and the middle is your bone. You see 23 years old men have very beautiful muscle. And during aging, 78 years old men, you see a lot of muscle loss, the fat and the collagen inside your muscle. And progressing loss of muscle, progressing loss of independency, it become a very important issue for elder population because you will lose your independency and lose your good quality of the life. But what kind of factor can actually regulate your muscle aging? Can be your lifestyle, hormone, medication, and so on. In my lab, I'm interested about impair of the muscle regeneration. We are looking for anything that can restore the muscle regeneration capacity because we know during aging, our muscle regeneration capacity are dramatically decline in the elder or in your age muscles. And for study the muscle regeneration and muscle aging associated process, my lab focused on the three different directions. So one is study the RNA regulated network between RNA and RNA binding protein to see how they function together to control the human myogenesis. Another one, I will introduce non-coding RNA a little bit later. Another one, we will focus on the organelle non-coding RNA function in muscle aging. As you might know, the mitochondria is important for the muscle contraction and also important for muscle aging. So my research will also study the non-coding RNA from the mitochondria of specific organelle and how they can regulate muscle aging. And a very vast part for my research direction, we will focus on the senescence, which we also call the cellular aging, and how they can regulate the muscle progenitor cell, muscle stem cell, and to control the muscle aging. And so with all these ideas, so we will very focus on some molecular marker, how the RNA control gene expression, and we try to identify some antisense oligo, potential RNA drug, and also some of the regulation function, and to see whether how the post-transcription regulation can be regulated by those RNA molecule to control muscle aging. The goal is to delay or restore muscle aging phenotype. And so in here, I want to give you a little introduction for the non-coding RNA, not too much detail, but I will also cover some of the example, which is my previous work. And we can use this work to also know how the non-coding RNA function is cells. So talking about a long-coding RNA, I will let you know about the human genome only have 2% gene have a transcript to encode mRNA. Most of transcript, most of gene in our human genome belong to the non-coding RNA. Also, long non-coding RNA by definition more than 200 nucleotide without protein product, and it can have a splicing poly-A tail, but it's poorly sequencing concept. But why long non-coding RNA is so important can be a potential marker in many disease models, and it might be regular important biological events. Because right now we also know that non-coding RNA usually has tissue-specific expression pattern or context-specific expression pattern. What's that mean? For example, in the muscle aging, This non-coding RNA may be only expressed in your skeletal muscle, but not any other tissues. And this non-coding RNA only highly expressed in muscle aging or during muscle regenerations. So with this idea, this non-coding RNA might be only fine-tuned very specific function to regulate biological function or physiological function and the disease model. I show some example from my previous research and just cover some of the non-coding RNA function inside the cellulose. Non-coding RNA can bind to mRNA or non-coding RNA can bind to the microRNA in the cytosolic to regulate the mRNA stability, to control the post-transcriptional regulation. Non-coding RNA can also inside the nucleus to control the transcriptional regulation by binding to the promoter region to interact with the transcription factor. Non-coding RNA can also inside a specific organelle like a mitochondria. In one of my projects, they will control the mitochondria encoding genome. Mitochondria genome is very complicated regulation, and little is known about how the mitochondria genome regulations. Non-coding RNA also have some function in other different organelles. With this all regulation, we are interested in study the non-coding RNA function in muscle aging and muscle aging associated process. One of my study I published last year. I will use the summary slide today. You know what we find in this research. So we found a non-coding RNA called OIP5S1. It will increase during the myogenesis, human myogenesis. And MIR7 is a microRNA. They will decrease during myogenesis. We found the non-coding RNA OIP5S1 and the MIR7 have very special pairing binding property. And this binding property will induce one of the very special machinery called target RNA-directed microRNA degradation. And this is the very first time people find in the skeletal muscle tissue, the non-coding RNA will induce microRNA degradation. And because this microRNA degradation, they cannot to target downstream regulation. So the muscle cannot fuse together. So we will reduce the muscle myogenesis. However, we also identify some of the molecule. We design some of the molecule called antisense oligo. In here, we call target site blocker. They can bind to this region to block these two RNA interaction. With this idea, this small molecule, kind of RNA drug potential molecule, the block interaction, they can modulate the muscle regeneration function. They have potential to get into the next step to study whether this antisense oligo can be functioned in vivo and in the clinical trial. And not only that, in our research, we only focus on different point of view to study the non-coding RNA. We were using something like this different approach. So we were using larger scale to do the RNA sequencing between different age muscles. We're using the muscle biopsy, the age from 20 to 80, cover a lot of people. And with using the RNA sequencing combined with different condition, we try to identify any candidate gene, candidate non-coding RNA or mRNA change during muscle age. And further down, when we select that non-coding RNA, we were doing the molecular mechanism study. We will study how this non-coding RNA really function in our skeletal muscle to regulate muscle aging. Say, for example, in here, we were using non-coding RNA to identify its enlightening protein. So we're using the biokinetic antisense oligo, pull down the complex, using mass spectrometry to identify protein. If this protein are function in mitochondria, the non-coding RNA can series protein to regulate mitochondria function to regulate muscle aging. So that will be the mechanism part. And the very last part, we will focus on the in vivo or ex vivo study. Say, for example, we're using the monkey biopsy sample to isolate progenitor cell from the monkey. So we found that the young monkey progenitor cell have a very strong muscle regeneration capacity. But in the age monkey, you will have less myogenic capacity. So they can do the muscle regeneration function is reduced. But here we kind of restore the non-coding RNA expression into the age skeletal muscle progenitor cell. They will restore the regeneration capacity. The idea is that the non-coding RNA can be a marker, can be a functional molecule, can be target potentially to regulate the age muscle, become the young muscle. That's the idea for our research. Try to identify those markers, study the molecule, target the molecule to restore the functions. So basically, we found a non-coding RNA that would decline during muscle aging by large scale human biopsy screening. And the reason why this non-coding ion decline is because when they have a lot of non-coding ion, they help mitochondrial function in the young skeletal muscle. However, in the old skeletal muscle, the non-coding ion level is low, so mitochondrial function is impaired. So the muscle regeneration function are declines. So this is kind of our research. And right now in my lab, we still have some cooperation in the US group in NIH and the other university still actively cooperate with those groups. And we also have some cooperation from the Taipei and also local. And so right now we are hoping to looking for some people from India, and they have a highly motivated and they were interested in molecular biology, especially RNA biology in aging or aging-associated disease model. So you can join the master or PhD program. So this is my contact information. And not only that, this kind of like an equivalent in my lab. And so we have a culture cell system to culture primary cell. We have also another different machine to study the gene expression, protein and RNA. We also have some flow cytometry and so on to study the function of your cells. Not only doing the work, we also take some time to enjoy the life and to discuss something. So we sometimes couple weeks or couple of months we have a happy hour together. This is me, and I just start my lab, so we don't have a lot of people yet, but we also have with another PI, so we have a couple of people together. We enjoy the food, sweet, and so on. But not only the food, we also can see the stunning sunset. This is actually the photo took from directly from my lab's windows. You see the sunset here. This is sea, so beautiful, and you must come to see by yourself. Then we also enjoy the tea time. Always discuss the science, but we also enjoy our life. And the final slide I want to thank for everyone, also my collaborator lab and also the local collaborator lab. And thank you everyone for your attention. And if you are interested, please contact me by email and phone number. And also you can search my name and the email in the website, and we have more information in there. And thank you very much.